right, everyone, here we are for bacterial growth part 2. In this part, we're looking at the growth of the population of cells, not just individual cells. One of the terms that we use often when we talk about bacterial growth is this term generation time. And so there's a definition for it over here, the time interval required for the formation of 2 cells from 1 cell, or the time it takes for 1 cell to go through binary fission and become 2 cells. And like I said in the last video, the time that this takes can vary a lot from cell to cell, in different living conditions, the right temperature, the not right temperature, a lot of things can affect it. One thing we can do is figure out how many cells we have in a population or how many cells we started with based on the number of generations that our bacteria are replicating. If we start with a single cell, we allow it to go through 1 generation, we know we're going to have 2 cells because we're going to double each time. 2 cells will then double to become 4 and so on and so on. And after the 5th generation, we'll wind up with 32 cells. An easy way to calculate or express the number of cells that are in a population after a certain number of divisions is by using the exponential value. So 2 to the number of generations. We could write that if we wanted to as 2g, and I'm using g for generations. We can then calculate, if we know how many cells we started with, we know how many generations it's gone, we can pretty easily calculate the number of bacteria that we have in our population at a particular time. So let's say that we start with 100 cells and we're going to let them grow for 10 generations. And we want to know how many cells we have at the end of those 10 generations. So n for the number of cells is going to equal n at the time of inoculation plus 2 to the number of generations, our exponential value over here. If we do that math, n equals 100 plus 2 to the 10th. We plug that in my calculator and we wind up with n equals 1,124 cells. We could do that backwards also if we wanted to know how many generations it had gone. We could do some different math to find out the number of doublings our population had gone through. It's a pretty simple math in the grand scheme of things. I mentioned previously that the generation time, or the time it takes to double, varies based on growth conditions. This is a chart showing us the amount of time it takes for bacteria to go through 1 generation when growth conditions are optimal. That means the best conditions for that particular set of bacteria to grow. On the really fast end, we have E. coli, which can go through the entire process of binary fission in about 17 minutes. That's pretty fast. A little bit slower is Streptococcus lactis. In milk, just regular old milk, about 26 minutes. In a lactose broth that's been formulated in the lab, about 48 minutes. Still really, really fast. Staph aureus, about a half hour. Now down here, we have some of our very slow growers. We have Mycobacterium tuberculosis, which causes TB. And it takes between 13 and 16 hours just to go through 1 round of binary fission. Treponema pallidum, one of our slowest ones, 33 hours. So these slow growers, we would not culture them for diagnostics because it can take for TB about 6 weeks to see a single colony. And nobody wants to wait for 6 weeks to find out if they have tuberculosis or not, especially when we have better testing methods. So those really slow growers are not going to be sent to a diagnostic lab for culture, whereas something like an E. coli infection would be, because you can get those results back overnight. When we start a new culture of bacteria, we can monitor its growth over time. And this can be really important if you're using bacteria during experiments because you want to make sure that they're growing really quickly when you carry out your experiments. And there are a few what we call phases to a bacterial growth curve. So there's 4 phases and they go like this. At time 0, when we put our bacteria into our brand new culture growing under optimum growth conditions, the first thing that will happen is not a whole lot. After a while, our bacteria will really start growing. They'll grow really super fast for a while, and then they will plateau off. And eventually, if we do nothing to this culture and we just let them keep growing, they're going to start declining. Each of these parts of our growth curve are the 4 different phases. 1, 2, 3, 4. The first phase is what we call the lag phase. Lag like lagging because not much is happening. The second phase gets called the log phase because the bacteria at this point are in exponential growth. They are growing as fast as they can. We say log because it means the same thing as exponential. The third we call the stationary phase. And the fourth, you could probably guess if you thought about it, this one is called the death phase. As a researcher, it would be important to know what phase of growth your bacteria are in at any given time because we want our cells to be in that exponential growth where they're happiest and healthiest to do experiments. So I'm not going to tell you what's happening to cells during all of these stages of the growth curve. We're going to talk about that next time we come to class. All right, that's all for bacterial growth and division.